So this is question 29. This is a discrete question, and it states which biochemical technique requires a pH gradient. So what this is asking is what biochemical technique requires you to have this gradient of pH across a certain distance, essentially. So A says limited proteolysis. Proteolysis is the breaking of peptide bonds. This does not require a pH gradient, so A cannot be correct. B, southern blotting. So remember, this is when you're separating on a gel. First, it's DNA, and you're separating by size, not necessarily by anything related to its pH. So southern blotting is not the correct answer. I'll move on to D, SDS page. So remember, SDS page, this is going to be separating out proteins after you denature them with SDS, and this separates by size as well. So this is protein. Similar to southern blotting, it separates out by size, except you're looking at protein. This does not require a pH gradient. So that leaves us with isoelectric focusing. What is isoelectric focusing? Essentially what's going on is you have a pH gradient on a gel here, and one side is positively charged and the other side is negatively charged, and you have a pH gradient. Different proteins have different pIs. What is a pI? That is the pH at which the charge of the protein equals 0, okay? Not that the pH equals 0. This is when the charge of the protein is equal to 0. So when you reach a pH of the pi, so when pH is equal to pi, the charge of the protein will be 0. So what happens here is you dump a protein here. Let's say you're just filling in the protein here. You dump in a lot of proteins. Let's say you just have a mixture of 3 different proteins. Yeah. So what's going to happen here is you're going to have a pH gradient from either low to high. And what's going to happen is, remember, when pH is greater than your pi, the protein's charge is going to be overall negative. When pH is equal to pi, the overall charge is going to be 0. And when pH is less than the pi, the charge is going to be overall positive. So what does that mean for us? It means that when you dump these proteins here, they're whatever pH they're at in the middle, they're either going to be negatively charged, positively charged, or in the unlikely case, they're going to be 0. So let's say in the first case, it's the pH is greater than the pi, it's negatively charged. It's going to actually move down here towards the positively charged side of this gel. It's going to get attracted to the positive side. It's going to travel, let's say it's the blue protein. It's going to travel, it's going to travel, it's going to travel until the pH at this point is equal to its pi. So the pH used to be higher than the pi when the protein was dropped off in the middle here, but as it moves to the left, the pH gradient decreases the surrounding pH, which means the pH is eventually going to reach its pi point. And what does that mean? It means the protein itself is going to have an overall 0 charge, and that 0 charge means it's not going to be attracted to the positive side or the negative side. On the other hand, let's say our protein here initially has a positive charge, okay? That means the protein is going to be moving to the right, okay? It's going to move to the right, moves, until it hits its pi point at a higher pH. Now the pH is equal to the pi, and now that charge is going to be 0 on that green protein, and it's going to remain right here. So it's a way of separating out proteins depending on their pi, which requires that pH gradient that the question asks for, making C the correct answer. This is the next discrete question. It says the gene for red-green color blindness is recessive and X-linked. What will be the expected phenotype of the children of a colorblind woman and a man who is not colorblind? So let's take a look. It says it's X-linked, right? So what does that mean? It's going to be a disease, or it's going to be a gene that's going to be living on the X chromosome, essentially. Remember, the woman is expected to have an XX chromosome, and the man is expected to have an XY chromosome. So here's what we know. This gene is this phenotype is recessive, and we know that the woman is colorblind, which means it carries the diseased gene, the colorblind gene, on both the X chromosomes. Now we know the man is not colorblind, which means this X has to be a wild type chromosome, essentially meaning it does not carry the gene for red-green color blindness. So the man is wild type and the woman has 2 X chromosomes that we know for a fact have to carry the color blindness gene. Now let's take a look at this with a simple Punnett square. So let's put the man on top and the woman here, and then let's just draw out a simple Punnett square. All right. If the man passes on its X chromosomes, the possible combinations are only one. and it's that you're going to have a daughter that carries the X chromosome that carries an X chromosome that carries the gene for colorblindness. But because this is a recessive trait, these daughters will not have colorblindness. However, if the man passes on its Y chromosome, well, we know that it's going to have to be the born child has to be a son that carries the colorblind gene in the X chromosome, and because it's X, these guys are hemizygous. The sons are hemizygous for the X chromosome, meaning that they don't have another X chromosome to rescue the colorblindness gene here. All of the sons have to be colorblind. So remember, for sons, they're going to be XY, and because they don't have another X chromosome, if they have a disease gene or some kind of gene on the X chromosome, that gene will show up. Whereas for females or for daughters, when they're born, because they have two X chromosomes, and in this case, one of those X chromosomes has that colorblind gene, well, because they have two, and as long as one of them is a normal copy, and we know that this trait is recessive, that means that this normal copy of the X chromosome can rescue and prevent colorblindness in the daughter. So just to reiterate again, sons, they only have one X chromosome, meaning if they carry a gene on the X chromosome, they have to express it regardless of whether or not it's dominant or recessive. Whereas daughters, because they have two X chromosomes, if they carry a gene on one X chromosome that's recessive, they will not express that trait because that trait will be dominated by whatever's on the other chromosome. And in this case, that trait is no colorblindness. So what does that tell us? That means all the daughters, all of the daughters, even though they carry the colorblindness gene in the X chromosomes, they themselves will not be colorblind. Whereas all of the sons, because they only have one X chromosome that carries the colorblindness gene, they have to be colorblind. So looking at our answer choices, it seems that B is the correct answer. Let's get started. This is the next discrete question. It says, which type of reaction has a KEQ greater than 1 and is kinetically fast? So let's take a look at the answer choices. We're looking at something that's exergonic versus endergonic, and high activation versus low activation energy level. So let's first address the first point, KEQ. If your KEQ is greater than 1, what does that tell us? That tells us that, let's say you have some kind of reaction like this, and it tells us that the reaction actually favors the products more than the reactants, okay? This reaction wants to go forward. So what that tells us is that in the forward direction, at least, this reaction is what? Spontaneous. And what's the criteria for spontaneity? Is that the free energy, the change in free energy has to be less than 0. And what does that mean? That means this is an exergonic reaction. So remember, exergonic means delta G is less than 0. Endergonic means delta G is greater than 0. Well, in this case, again, because we know KEQ is greater than 1, we know that this is a spontaneous reaction, which means delta G has to be less than 0, which means this reaction has to be exergonic. This eliminates A and B. Let's take a look at the second half of these answer choices, and we know that this is related to kinetics. Remember, KEQ, so the equilibrium constant, things with equilibrium, or delta G's, they do not tell you anything about the kinetics of the reaction. A reaction can be spontaneous, but it will not. But it's also possible that it's not a fast reaction. So, for example, let's say I have a spontaneous reaction, but it takes 1 million years for it to happen. So even if it happens, that means it's a spontaneous reaction, but it doesn't tell us that how fast that reaction is going to happen. So we know occasionally we think that spontaneity means speed, it's going to happen really quick. No, spontaneous means the reaction happens by itself, but it doesn't tell us anything about how fast the reaction itself will work. And so that's where kinetics comes in. This will tell us how fast a reaction happens. Now, if it's kinetically fast, let's take a look at a graph. What determines the kinetic speed, the speed of a reaction? Let's say I have this is a free energy on the y-axis, and let's say I have a reaction progress. If I start off with the reactants up here, and let's say I come over to this little energy hump, and then I end up with products here. So we know that this is actually what's happening, at least in this case, because, like I said, it's an exergonic reaction. Delta G has to be less than 0. So we see that drop in energy, which makes it a spontaneous reaction. However, kinetics is very worried about this difference right here. What is this? This is the activation energy. And you can think of running these reactions like someone rolling up a boulder up a hill, so let's say if this hump is very large, it's going to take a long time for that person to get over that hump to finally get here. But if this hump is smaller, the smaller it gets, the easier it is for that person to just roll that boulder up the hill, right? In the same way, if the activation energy is smaller, that makes this hump smaller, meaning it's easier for reactants to become products, thus that reaction is faster. So if we know that this is a kinetically fast reaction, we know that this activation energy, this hump has to be small. So we know that it cannot be a high activation energy level. It has to be an exergonic reaction with a low activation energy level. Let's take a look at the last question of this discrete set. It says, blood glucose levels in a healthy individual as a function of time are shown. In response to changing glucose levels, which events occur at time points A and B? All right, so we'll take a look at A. We'll see high blood glucose levels, and look at B. We see low glucose levels. So what's going to happen there? Well, let's take a look at answer choice A. It says hepatocytes secrete glucagon at point A, and the pancreatic alpha cells secrete insulin at point B. Well, here's the first problem. Hepatocytes, so hepatocytes are liver cells, okay? Liver cells don't secrete glucagon at point A, okay? It's the pancreas that's going to be secreting glucagon. So immediately, A is an incorrect answer. Not only that, the pancreatic alpha cells, these are not the cells that secrete insulin. It's actually the beta cells, okay? The beta cells that release insulin, not alpha cells. So they have this wrong on both sides right here. So A is not the correct answer, and let's talk about a little bit what's going on in this problem. Let's say we have a blood vessel, and let's say in this blood we have a lot of sugar molecules floating around. If we have a lot, our body's going to want to get the sugar out of our blood and into the cells that need it. And what's the key? What's the key that gets the sugar into, the glucose into our cells? That's going to be insulin. Okay, so the body releases insulin when there's a lot of sugar in the blood, and it tells the body, okay, we need to get rid of this sugar, we need to get this sugar into the cells in the body that need the sugar. And how to do that? We use insulin. We release the insulin as the key so that the sugar can get into those cells that need it. So insulin is released by beta cells. Let's think of a different scenario here. Let's think of a scenario in which your blood doesn't have a lot of sugar. Let's say, you haven't eaten in a while, and your blood sugar is very low, and there are other cells in the other parts of the bodies that need this sugar. Well, how do we release sugar into the blood? We use something called glucagon. Okay, glucagon is developed by alpha cells in the pancreas, and glucagon is going to tell your sugar stores, the cells that store your sugar, hey, we need to release some glucose into the blood so that other cells can use it. And so that's where glucagon comes in and says, okay, let's break it up. Let's break up these glucose stores, and let's release that glucose into the blood. And that's glucagon being released by alpha cells that mediates this process. So let's take a look at what's happening. So in A, we see high blood glucose levels. So we're fitting description 1 here, where insulin is going to be needed from beta cells to get this sugar out of the blood and into our cells that need them. B shows us low blood glucose levels, which corresponds with our scenario 2, where glucagon is going to have to come in and release sugar into our blood to get to other cells that might need it. So let's take a look at the answer choices that fit our description. So answer choice B says that at point A, we're going to have pancreatic alpha cells secreting glucagon. So pancreatic alpha cells do secrete glucagon. Remember, when we have high blood glucose levels, we don't want more glucose in our blood. Our body's not going to do that. We want to get that glucose out of the blood. So we're not going to secrete glucagon, we're going to secrete insulin. C says pancreatic alpha cells secrete insulin at point A. Well, that's incorrect. Remember, alpha cells don't secrete insulin. It's beta cells. So that leaves us with D. It says pancreatic beta cells secrete insulin at point A, which makes sense. Insulin is going to be secreted by beta cells to get that glucose out of the blood into the cells. And pancreatic alpha cells secrete glucagon at point B. So we have low blood glucose levels. Then we're going to have the alpha cells secreting glucagon to get glucose into the blood to other cells that might need it, making D the correct answer choice here. I did want to clarify one thing. I think I said earlier, insulin is released by beta cells when there's too much glucose in the blood so that cells can absorb that glucose and use it. Rather, it's actually more clear to say that beta cells release insulin so that glucose can enter cells and be stored in the cells when the rest of the body doesn't need it. So if your blood has a lot of glucose, Your body's like, okay, we need to store this glucose somehow. We're going to store these glucose molecules in our cells in the body. And how do we do that? We have to get the glucose into those cells. How do we do that? We release insulin from the beta cells so that insulin can act as a key so that glucose can enter those cells and be stored in those cells for use later. So just want to clarify that point on this question.